Let's invite Dr. Professor, the almighty, the legendary.
Sorry, one of my three-legged cats is pushing the dog bed around the hallway. My name is Thomas Merritt. I'm a professor in the School of Natural Sciences at Laurentian University. I've been here for about 17 years. I run a lab looking at the genetics of stress response, mostly, using fruit flies, Drosophila melanogaster, as a model, which is how Balint and I know each other. In the last, actually longer than I thought it was, but say 5 or 6 years, I've started doing more science outreach and writing about science. And so I do a fair amount of talking about science. In general, if I had to tell people that I would like to talk about the genetics of stress in fruit flies, get a lot of blank stares. But if I say, hey, do you like coffee? Then 80% of North America says, I do. And I'm like, would you like to know more about the biology of coffee? I would. And so that's been my hook for science communication. I'm going to start with a teaser. I think what we should talk about next time, assuming this goes well and you invite me back again. Right? Can you imagine? Like, how bad would it have to be for you to say no?
What
right?
you do with, like, after Thomas? Everyone like looks forward to seeing you. It's always, where's Dr. Coffee? Where's Dr. Coffee? I can't imagine.
So let's go, right? But it'd be sort of fun. Like, where's the line? Okay. So assuming that we get this to, this goes well for the next 50 something minutes. Next time, let's talk about species. So there's some really, I don't know if you've talked about species and speciation. It's one of my favorite topics to piss off people who work on species. Because species
for every Friday we review a paper called for our species of the week. Ooh. And so every week there is the debate on, well, is this sufficiently enough a new species to call it a new species, and are species even real?
Okay, species are not real, and that is the starting point. Their species do not exist.
So we would love to have you on to chat about that, because it is something that is commonly debated here as well, because like on these different papers, even if it's sequencing, the threshold of what is new species with the sequencing data is different between papers. And so there's just, yeah.
Species are an artifact. They're a really convenient way for us to break up the world around us. They're not real. And so I say that to piss off people who work on species 100%. It is actually true. So you can point at a mitochondria and like, that's a mitochondria. You cannot point at a species. A species is a concept. It's an abstract concept. It's an artifact. It's a way that we define the world around us. So there's some really cool stuff in coffee species. And so we could get back and chat coffee. So maybe next month we'll shift from Mondays to Fridays and we'll do like a coffee species chat. So leaving that. So remind me that one of the last things to chat about is what the stuff we're going to chat about today says about species.
Okay.
So that's who I am. So I'm Thomas Merritt. I'm a professor. I do genetics, mostly with flies, and I talk about science. So we were talking about porcupine sex toys last time.
Yes.
And somehow we segued from that to, let me tell you a cool story about devil face cancer, right? And there must have been some connection in there. Okay. So
I don't know what the connection was.
So I think the connection is the Tasmanian devil facial cancer is seen as a sexually transmitted disease, okay? And it is actually a sexually transmitted disease in a very atypical way. So the disease, and it's abbreviated DFTD, so devil facial tumor disease, and it is horrible. So it looks terrible, and basically what happens is the animals break out in these little tiny lesions. The lesions get really big, really big, really big, really big, to the point where the animal can't feed and it dies. The fatality rate is essentially 100%. The transmission rate is essentially 100%. And so this was described, I remember reading about it in sort of the early-ish kind of 2000s through the 2000 teens-ish. And it was really described as the end of Tasmanian devils. So Tasmanian devils are these marsupials that live in Australia. They look really vicious. It ends up that they're not particularly vicious. They look like, they actually look a lot like a black possum. So if you picture a possum, slightly more fierce, fuzzy tail, and black instead of the various grays of North American possums, that's a Tasmanian devil.
And a little bit of their sounds too. So we were chatting a little bit earlier that they make these scary sounds that when Westerners first came to the islands, they were just heard it at night, and that was one of the naming conventions as to their Tasmanian devils because they sound scary.
That's really cool. I didn't know that. Okay, so now, tit for tat, you've taught me something. I'm going to teach T cells. So the reason this is considered to be a sexually transmitted disease is that the males fight each other for access to females, and then the males and females are actually a bit bitey during intercourse, and they're not a bit bitey. Apparently, they're like really, really, really bitey. And so what happens is the animals are basically injuring each other in the face, and then these injuries, instead of healing, blow up into these tumors that kill the animal. What we realized 10, 15 years ago was that it's actually this cancer, and what's happening is it's being spread across the population. And so it took a while to figure this out, because like many of the things that we've talked about in the past, this isn't how things work. And so the reason that I wanted us to chat about it is this mechanism or etiology of disease transmission is almost heretical and is now completely embraced. But traditionally there are, and I had to write this down because I'll never remember them off the top of my head. Traditionally, there are 4 modes of disease transmission. You've got, and so, okay, guess. What are the modes of disease transmission?
Reproduction. STDs.
What kind of reproduction are you doing?
STDs.
No, like how do you, sorry? STDs. No, sorry. Okay, but what's the actual disease mechanism there? It's not reproduction. That's not why we're doing it. Okay, I'll give you a hint. One of them is viruses.
Bacteria.
Okay. So viruses, bacteria.
We have from the chat air and blood.
So, but so yes and no. It's being transmitted through air or blood or saliva, but what is the it in that sentence? So what's being transmitted? So one of the other 3-legged cats is coming to say hi. What's being transmitted is either a virus like HIV, a bacteria like syphilis, or fungi. So disease, the other vectors are bacteria, virus, fungi, and then parasites. Okay, so that's typically how we think of disease. The devil story has made us rethink that, because what's actually being transmitted is the cancer itself. And so the cells are being transmitted. They infect an individual, and then that infection blows up. One of the things that we actually still don't know is which direction the infection's going. So it's not clear. Basically, there's so much violence in Tasmanian devil, either the males fighting, or it looks like it's the sex itself, that it's unclear whether the uninfected are biting someone that's infected and they're getting infected, or the infected is biting somebody and then passing that infection on.
So it's unknown whether you're delivering through maybe the saliva versus the blood?
No. So that's the thing, is it's actually 100%. It looks like it's maybe a combination of the saliva and the blood. It's just it's not clear who at the start of this wasn't infected, because both parties are biting each other so extensively that it's hard to tease apart who's getting bit. Everybody's getting bit and everybody's receiving bites. And so it's unclear exactly which direction. But what happens is that at the end of this sexual intercourse, both individuals now have cancer cells that then spread.
So I wanted to
talk
about Mama_Izu_fan just
also,
subscribed
Mama, thank you for that
25 months.
Tier 3 resub. Thank you, Mama, as a fan. 2 questions for you. One is this virus anything like HPV, where the virus itself is causing cancer? And then 2, are those injuries, do we know if they're superficial between the 2 individuals, or if there's more of a deep cut in there, like something like in the lymphatic system or in the sinuses that are also involved?
Okay, I'll answer the second one first. I think the cuts are actually pretty severe, like they're bites. And I don't know, I didn't actually read anything that says whether it's a skin transmission. I think it's actually the tumors are okay, I don't know, but we can fake it. So what we do know is that the tumors start off in the face. So you don't get a bite and then you end up with a tumor in the lymphatic system. You get a bite, you get a facial tumor. Eventually they do spread and the tumors end up systematic, but they start with being localized. The thing that's really messed up with this Tasmanian devil cancer that's made us rethink disease is what's being transmitted is the cancer. It's not a virus. It's not a bacteria. It's not a fungus. So when I was actually talking to my girlfriend, we were out for a hike over the weekend. I was like, yeah, I'm going to talk about Tasmanian devil sex and disease transmission. And it's a lot like HPV, because most people know about HPV because we started vaccinating teenagers to prevent HPV. So in HPV, and there's actually another human cancer that's the same thing, Do we need to do the raid thing?
Oh,
I just saw a raid.
Well, we're Gucci. We're Gucci. I don't want to cut you off.
Okay.
So you're rolling.
I'm on a roll. Okay, so the thing that with HPV and other infectious viral-driven cancers, what's being transmitted is the virus. And if you tried to transmit the cancer, it would actually be recognized as foreign and destroyed. So HPV is a virus. If you've had 3 sexual partners, your chance of having HPV is essentially 100%. A subset of HPVs lead to cervical cancers. And what has been demonstrated really clearly is if we use vaccines to knock down HPV, we almost eliminate, and in some cases, eliminate cervical cancer. It's a super cool medical story. But it's a little bit unusual in that the cancer is being caused by this viral vector. So usually in cancers, you get a mutation in the body, a somatic mutation. That mutation leads to misregulation of genes and a blow up of cell growth. In these viral cancers, what's driving the cancer is actually an infectious agent, a virus. So the HPV story caused us to rethink how disease worked, because we think of cancer as being something in your body. Maybe you've got an allele that predisposes you to it, but it's a mutation that happens here. In viral-driven cancers, what's driving the disease is actually the virus. So there's a virus that infects the cells. That viral infection messes with gene regulation, and you end up with a tumor. Okay, so those are 2 different paths to cancer. The devil story is a third path. What's being transmitted
is the cells. So mutant cells are moving. So that's a wild way to transmit cancer.
And so why is this important? It's important for a number of reasons. One, we don't want to lose any species. So species diversity is super important. And it looked like Tasmanian devils were going to go extinct because there was this cancer just ravaging through the community. And we can talk about why it looks like that's maybe not happening now. But B, we need to understand disease. And so there are actually so one of the interesting things with the devil cancer is there's now a second devil cancer. So if you picture what we have just described, every devil out there with cancer has a cell from the same lineage of cells. There was an individual that had a mutation that created a tumor that is then that tumor spreading through the population. And as of about 2006-ish, it looks like there was a second devil cancer that's spreading. So we had this one event.
That means that in 2 instances, this happened and has spread throughout the population. And so what is the cell itself that is being jumped? And why can that cancer be transmitted so easily? Like, why is it that we can't, or I don't know, maybe we can't just transmit cancer to one another through blood-to-blood contact?
Yeah, so usually there are a series of, we shouldn't be taking notes because we're going to lose all this. There are a series of things. So one, the 2 things means they're two independent events. B, we don't know what the cell is because part of becoming a tumor means that you don't look like what you used to be, right? So I was thinking about this too, like, can we say what the cell was? I don't think we can. We literally could tell you the cell came from Bob, right? Because it's actually this lineage that now the lineage is mutating through time. And so it is changing. If you think back to when HIV was just like spreading wildly, we had all these different strains of HIV. There was an initial HIV infection, and then those infections spread in much like what you might call a species, except they don't exist. Okay. So the cancer tumor, there's diversity within that tumor, but it's just mutations that have accumulated since this tumor was initially established. There's a really great question of why this doesn't happen all the time. And most cancer cells are actually recognized as foreign and destroyed. And there's something about this facial devil tumor that the cells are not recognized as foreign and destroyed, and instead they grow. But what's growing is actually this clone. It's not. So typical tumors, it's you gone crazy. In this case, it's a parasitic lineage of cells creating tumors throughout the body. So we need to understand this.
So this lineage of cells, and when we talk about tumors in the past, right, we go and look and see like there's the mix of cell populations, right, within a given tumor. And that heterogeneity or that mix makes it really hard to tackle because there's so many different cell types that could be mutating. So are you telling us here is in this instance it is a clonal cell type? So are all the cell types within a patch of face tumor and the devil identical to one another, or are they also different and constantly mutating as well?
That's a great question. I don't know the answer. And what I can tell you just as a geneticist is I'm assuming there's got to be diversity just because mutations occur through time. But I didn't actually find a paper, not to say that they're not out there. I just didn't even think to look for it. So I haven't found a paper that says what is the diversity of cells within that lineage, or even what's the diversity of cells within the different lineages. So this virus has been around for a couple of decades. Presumably they're different lineages. But what I can tell you is, since we know there's a second lineage, there must be little enough diversity in the first lineage that we can say this is a discrete lineage. But there are lots of really interesting biological questions around why is this cancer successful in the face that most cancers are not. There are some cases of human-to-human transmission of cancers, usually in immunocompromised individual. And so there'll be something that moves a cancer from patient A to patient B, but the patients are already immunocompromised, and so their body isn't recognizing the cells as foreign and destroying. There are at least 2 cases of transmission in non-immunocompromised individuals, but in neither case did the tumor metastasize. So there was a surgeon that basically inadvertently inoculated himself with a cancer with, I think, a scalpel, and got a local tumor that was excised and didn't go anywhere. And there was another case of a needle prick where somebody was pricked by a needle that I think had done a biopsy on a tumor, and they got a local growth that was able to be excised and didn't go anywhere. So far, we're not seeing spread of similar cancers in humans, but the fact that we have seen it in one organism, and if we're all fundamentally related, means that we need to understand this so that if it does, we do see something like this in humans, we know what to do about it. Does that make sense?
The immediate human connection is a future possibility of how disease transmission could work, and even though we haven't seen it yet, doesn't mean that we won't see it at a later point.
Yeah, and what's especially frightening is this Tasmanian devil facial tumor cancer isn't the only case we have. So there are at least 5 different clams, mollusks, that have transmissible cancers. These are actually passed through seawater, so it's not physical contact, and have wiped out populations.
That would be crazy to spread through that. That's like cancer in the air.
Yeah, and have wiped out populations. So it can happen in other cases. The immune system in invertebrates is pretty wonky. I don't know how much, when you were doing fly stuff, you looked into it.
That was my undergrad.
Seriously?
Yeah.
There you go. Okay. So, I mean, there's an immune system, but it's not the adaptive immune system that we see in vertebrates, right?
Like with, for folks in the chat, like with T cells, B cells, it's not that in invertebrates. Instead, it's rather the immediate like phagocytosis of eating bacteria and popping off large organisms and sticking them off to the side of the open circulatory system.
So maybe we don't have to be as worried because those are just mollusks. Okay. Except the classic case of transmissible cancer is actually in dogs, and was described it was experimented on in the 1800s. So there is a sexually transmitted dog cancer that is found globally, and it's essentially a tumor cell that's transmitted by genital contact in dogs. And
is this limited to a breed, or just like any dog can pick this up in theory?
Yeah.
Wow.
Yeah. So in theory, any dog can pick it up. And I think the deal is it's basically rare and identified, and either the animal's euthanized or it's excised. But we've studied this since the 1800s. Here's the punchline. Recently, we've done genetics on that cancer, and it's a clone. So there is a single individual, but it's the same game as this devil. It's the same story, right? So there was a mutation in a cell that created a tumor. That tumor is unique in that it can be transmitted. This should sound familiar because it's exactly the devil story, but now in a mammal instead of a marsupial. So if it can happen in dogs, it can likely happen in other mammals, including the world's worst mammal. So
which is
this kind of us.
Okay.
I mean, at least as far as like ecological impact and stuff. So, I mean, that's not even like subjective. Like if you look at what mammal has had the greatest negative impact on the globe, hands down, that's us. Okay. So if it can happen in other mammals, it likely can happen in humans. We need to understand how. The really funky thing in the dog story is when we look at the genetics of that lineage, it's estimated that initial mutation happened between 10 and 12,000 years ago.
And it's been maintained to the point where you can see it's a single lineage. So does that mean there's not really any, I mean, presumably, like you said, there is some heterogeneity or some mutation that's happening within these. But if you can trace it that easily, there must not be a really strong pressure for it to change very rapidly or to come up multiple times, right? It just seems to have been very very successful as a parasitic cell line from the beginning.
The second half of that is absolutely true. This has been very successful as a parasitic cell line. I don't think the first half is. You can, with reasonably simple phylogenetic tools, you can follow a path of change. And so the fact that we can estimate, given some kind of approximation of a molecular clock, how old this thing is, just means it's been accumulating change over time. Not that there's really tight constraint that doesn't allow it to go off in different lineages. I don't study this field. I haven't looked at the genetics all that closely. I wouldn't be surprised if there are lineages within that. But it's similar enough that our best guess at the moment is that it's a single isolate that's been floating around the population. So this is like the risk of giving away the speciation punchline. What's a species? Tumor cell is canine. Tumor cell is genetically a dog, but it has been a tumor cell for 12,000 years. So if you, like, genetically, it's a dog. Phenotypically, it's a tumor. I'm the only one who think that's amazing. Okay, so I will say in your defense, when I explained this to Tammy, my girlfriend, earlier today, she's like, oh yeah, and sort of did one of those, much like the look that you're giving me at the moment. But it's like, basically, these two things, one, this undifferentiated mass of tumor cells, and two, tiny, that was sitting behind me, are genetically almost identical, but they're completely different phenotypes. One's a tumor, and one is a dog. Okay, I thought that was cool.
So,
all right, if it lands flat with you, that's fine.
Back us up, Thomas. Back us up a little bit. What should we be visualizing when seeing that? So are you meaning to say that it's almost like an epigenetic difference in terms of what the cell type regulation is? Like from this tumor, could we make a 12,000-year-old dog, essentially, like the lineage was?
Yeah. No, they did that. They actually cloned the dog in the lab.
Wow. Okay.
No, I'm making that up. I totally made that up. Totally made that up. But wouldn't that be cool? They're like, okay, this is Betty. Betty's mom's a tumor. No, they didn't do that. But what I'm saying is, if I gave you a DNA sample and I said, hey, Balint, what's this? You would run it through a DNA sequencer and you'd come back to me and say, it's a dog, sort of a messed up dog, but it's a dog. But it's not a dog, it's a tumor.
So is that functionally different than if I were to take a cancer cell from a modern day human patient and compare it to a regular human cell? Would you have the same kind of appearance of like, oh, look, it's two humans? Or would it be very much like cancer and human?
Great. No, 100%. You'd have that same overlap. The difference is this tumor has a lineage as a tumor that is 12,000 years old.
So for 12,000 years, this cell line has been existing inside of a dog as a tumor.
A series, not one dog, just to be clear. We don't have a 12,000-year-old dog, but a series of dogs.
So it's been evolving. We have essentially genetically a 12,000-year-old tumor. So why? Why do we have this? Like what about it makes it, right? Because if we're thinking about it too, in terms of when humans are going to be affected, right? What is it special about the dog and the Tasmanian devil that facilitated for something like this to mutate and to evolve?
Yeah.
And are we special? Or is it going to happen to us too?
And so B, it will happen to us. A, we're not special. So you have to be really careful though with why questions. So there is no why, because a why question implies like a destination. This change happened so that this could happen. There is no destination. It's just that this change happened, and the impact of that change is this immortalized cell line. It's unusual in that this immortalized cell line is transmissible. Most immortalized cell lines die with the host, right? So I get a cancer, and when I die, that cell line dies. The unusual thing with these two lineages within the devil and this one lineage within dogs is that tumor didn't die. And it's not immortalized because we've kept it in the lab and we've been doing funky things to keep it alive. It is literally parasitically moving from host to host to host and maintaining that tumor existence in the case of the dog tumor for 12,000 years.
And in the case of the dog and the Tasmanian devil, there is a negative fitness effect, right? So there's nothing beneficial for having this. There's nothing mutualistic. There's still a decrease in survival rate, right? So is it truly a parasitic mass of cells?
And so that's what I was going to, correct you in the sense that there's absolutely a benefit to the tumor line. There's just not a benefit to the host. And what has to happen basically is infection can't compromise the host to the point where it can't be transmitted if that lineage is going to continue on. So in the dog case, whatever is happening, and I didn't look into the overall impact, but I think that the final impact is bad. Like I don't think that the dogs walk around with little like genital warts or something. I think they get tumors and die. But the impact happens after they have then spread that disease.
So
okay?
it's they spread, but so when they're sexually active, they're presumably not showing signs of the symptoms, maybe, of tumors, but the cells are there. The cells get passed, and then maybe they end up dying. So maybe something like these Tasmanian devils too, where you could have something like a low-level tumorigenesis, you have a mating event or the male-male aggression right over food resources, and then those wounds allow the cells to pass. And presumably, like the ones that have huge faces wouldn't be partaking in like the fighting anyway, because you'd be really sickly.
Yeah, and so there's going to be at some point where the tumors are going to take over and you won't be able to fight. And I think I got this right when I described it. The fighting is actually between males for females. So the misnomer with the devil thing is that most of their life cycle is actually pretty low key, but they are very violent when males fight each other for access to females, and then during copulation, the males and females both bite each other. And it looks like the transmission of the cancer is actually during that male-female copulation. So it's actually during the sex that fighting leads to transmission of the cancer.
Do we know what is, or if there is a certain cellular number of cancer cells that you have to transmit during this fighting to take? Or is it where maybe one is sufficient because they just don't seem to be recognized by the immune system? Like it's a very interesting transmission story, right? Of like, what are the thresholds that you need to get the disease state?
Yeah, and so what we do know is the threshold is very low. And so this is the story that I remember from maybe 5 or 10 years ago, was that essentially it was moving, the cancer was moving through populations of devils unchecked. And so it wasn't like a rare event, it was moving pretty quickly. And then the other half of that was that once you were infected, it was a death sentence. And so the tumors just got bigger and bigger and then spread systematically until you died. So and what happened to devil populations was they crashed.
So why do we still have devils?
Well, there are a couple of answers to that. So, sorry, the three pieces are everybody seems to get in, everybody seems to die, and the populations are crashing. And so this led to a lot of discussion of what are we going to do to save this species. Okay. So, sorry, you just asked me a question, and I was too busy waiting for me to tell you those three things that I didn't process the question.
No, so we have more questions in the chat as well. So before we get there, so does it, from Coffee Rocks, does it spread from mom to pup? Or is it, as far as we're aware, it has to be like during these like facial wound battles?
So that's an interesting point. In this case, everything that I read is that it's a facial wound spread. There are some cases of cancers being spread from mom to infant in humans, not in devils. So there are human cancers that are spread. They're very rare, and so far all the examples that I could find were one or both of the individuals were immunocompromised. But that's a really interesting point. So that, because there is this blood connection, that disease transmission route is something from mother to child is something that we are very cognizant of. And there are some cases of transmissible cancers that have been transmitted in that way. But that's not the case in the devil stuff.
And then do we know when this started happening in terms of the Tasmanian devils? Like, how modern is it? And then to get at your question, like, we're seeing this happen, how do you decide, or how does a scientist decide when it's time to intervene? Or if it's something where this is a natural event that has occurred, right?
Yeah.
Is it number one? And then at what point is it important for us to intervene on something like this?
Yeah. So those are really great questions. So the initial description of the condition was 1996. The description of the second lineage was 2006. So we've got a timeline on that. And my understanding is the crash of the populations correlates with that 96 initial description. The question about when we intervene is super cool. So it's actually super, super, super cool. Let me give up a plug. There is a book called How to Love a Forest by Ethan Tapper. And Ethan's coming here to Laurentian tomorrow to give a talk in my girlfriend's is the director of the School of Architecture, and every year the School of Architecture reads a book, and they have one school, one book. Super cool. Also really cool. The last 2 years, the book has been my suggestion. Tammy doesn't broadcast that. I'm not an architect, so but we're all reading Ethan's book, How to Love a Forest, and Ethan's book is really well written. He's a young guy. He writes really, really, really beautifully. The point of his book is that we can't not intervene, and so he's a forester, but he's a restoration ecologist. He bought a couple hundred acres of really badly impacted forest in, I think, Vermont, and his initial thought was, I'm going to let the forest heal itself. And the conclusion that he came to is that we have messed up the planet so badly that we can't just not intervene. And so when we finally drive ourselves extinct, the planet will go on, and it absolutely, the planet is going to outlive human beings. But while we're here, we have such an impact on the planet that we almost, if we want things to be maintained, we almost can't just leave it alone. It's really problematic because, the way that we intervene often leads to worse problems, but simply doing nothing can also lead to problems. So Ethan's book is a really great read on why we should be actively engaged with healing things, and why, given the state of the world around us, we can't just say, I'm going to let the forest heal itself. Okay, so in this case, I think the decline of devils probably goes back to Cook. And that's not even true. The decline of devils probably goes back to the initial landing of modern humans on Australia. So as soon as humans hit any place, they start to modify the environment around them. So devil populations are really not sustainable because they're competing with humans. There's habitat fragmentation. We've introduced mammals, so not marsupials, that are out competing them. So the devils are already at a bit of an edge, and now we get this facial cancer that comes in. The interesting twist is that the last thing that I sent you, the conversation piece, is there's some really good, essentially, epidemiology that suggests that Tasmanian devil populations have flattened out. And so they definitely crashed when this cancer hit, but they seem to have flattened. In addition, what seems to be happening is some individuals, the tumors are now shrinking, which implies that the population is going to start to recover.
Does that represent some type of immunity towards these cell lines, or what do we know? Where is that representing?
So they're working on that. What it does seem to represent is resistant genotypes. And so we've got some set of genes that are involved in resistance to this cancer, and there seem to be genetic diversity within those, and certain genotypes that are more resistant to others. And what's happening in the population is that we've essentially weeded out the individuals that are really susceptible, and what's left in the population are individuals that are more resistant, and they're beginning to increase through time.
So that's not from human intervention. Is that just from natural dynamics of populations?
Exactly. And one of the points that's made in that article that's really cool is right now, the approved human intervention is to captive rear naive devils, so devils that haven't seen the cancer, and put them back into the population so that we're increasing numbers of the population. And what the authors point out really astutely is that those individuals are going to be highly susceptible to the cancer. So what we should be doing, it's just really hard, is finding these individuals that seem to be resistant, breeding them, and introducing the resistant devils back into the population. So it looks like the human intervention could actually be working against the best interest of the species.
Is there any benefit that you see on increasing the population of susceptible devils?
The argument is no.
Okay.
Like, basically, because this virus, that's where these things come from. Because this infectious cancer is so infectious and so deadly, if you put an individual that's not resistant, it's going to die. And in fact, what it could be doing is spreading that cancer through the population more effectively because it's very susceptible to it.
So it's not the devil breeding that's difficult. It's pulling in the devils that have this resistance. And then, like, the breeding part is fine. It's just identifying the ones to breed.
Exactly. And so the paper that I sent you is within the last couple of years. Basically, this idea that the devil populations are rebounding is so new that information hasn't made it into the idea of how do we supplement devil numbers effectively, we didn't realize that there were any devils that were actually evolving resistance over time, or any members of the population that had resistance to that cancer.
Do we know if the cancer can spread to other mammals, or is it, I think this is kind of getting back to the original rabbit hole, right? Like, what limits the spread of this kind of cancer? Is it only to these devils? Can it spread to something that's closely related in terms of marsupial? Like, close relative was the thylacine, that's extinct, but maybe something like a koala, which is a close relative of.
So, ironically, there's actually quite a bit of koala devil intercourse that goes on. It's one of the unexplained mysteries. Totally making that up. No. So, as far as I know, can you imagine? You're like, everybody's like, seriously, I'm picturing this cute little koala. Like, okay,
look, the way when you deadpan at me saying that, all right, I'm like, wait a minute.
So, Chihuahuas and Great Danes, apparently so who knows? Like, we're not taking anything off the table. But I don't think we have any examples in any of the cases that I've given you of this kind of infectious cancer crossing species boundaries.
At least naturally. So do we, and presumably I don't think this has been done. Like, I tried finding something, but taking some of those cells from the Tasmanian devil and just implanting it into another organism. Like, will they?
Yeah. So could we create face cancer in possums?
Right, artificially introducing it.
I haven't seen that. I don't know if we even have things like marsupial cell lines. I mean, that's the kind of thing that I think everybody would hesitate. Like, I don't know. As far as I know, nobody.
So there are opossum cell lines because apparently they're highly cancer resistant. That's one of apparently the great reasons to study opossums is because of that. Actually, the only reason I know that is because my father studied opossums for that reason.
Are you making that up?
No. He ran his lab at.
My father was in fact a possum geneticist.
Mine was a cancer geneticist, but he worked on opossums, and he was part of the team that sequenced the original North American possum genome. So maybe they were on the same paper.
Yeah, my dad was just a guy from North Carolina, and so at some level they're all into possums. So everything else I made up.
Y'all see, he gets me every time. I'm like, really? Oh my god, they made a bit on the same paper.
That's really awesome. So no, I did not know that about your family history, and there are some weird lines, some unusual species with unusual patterns of cancer resistance. I didn't know that possums were on that list.
But it would be interesting to see either way if there's any kind of lineage. Like, if you were to implant it in a mouse, maybe they're too diverged to take. But I would just be curious. Like, obviously the koala, as you said, isn't getting frisky with the Tasmanian devil. But maybe if you were to do like blood to blood contact with them, or even on cell lines, if there's a way to be pulled in, and just like spreading for physical barriers isn't happening. But if there was to be some kind of spreading, like, how widespread is something like this? Is it just a generic mammalian cell line that's gone rogue, or does it have to have some genetic root in the host because that's how it evades the immune response?
Yeah, and I mean, we know that diseases change through time. I mean, we all lived through the most recent plague that at some level involves movement of a virus from a host to a new host, right? So that kind of thing is possible. I don't know of anything in the cancers that we're talking about. My understanding of what understanding we have of the facial cancer is that the reason it's not recognized is somehow this cell line looks enough like every Tasmanian devil that it's not destroyed by the immune system. And so the fact that it's going from devil to devil isn't a coincidence, and isn't simply because devils are only having sex with devils. That it's that relationship, the genetic relatedness, the reason that cancer still looks like a devil, even though it's been a tumor since at least 1996.
And then the same thing in dogs for several thousand years now, which is, again, it kind of gets you to the question of what is the immune system looking for, right? Is it a signal that, hey, I'm dog cell, or I'm Tasmanian devil cell that needs to be there? Or is it an absence of a certain signal, or is it both signals? Yeah. And then it's a very complicated cell line and cell structure that's able to be transmitted like this.
And disease is an arms race. So the reason that HIV took off was that HIV was able to move around the immune system and infect cells that were normally involved with killing off or destroying viral particles. So you just need a mutation here or a mutation there, or a pair of mutations that allow whatever this infectious agent is to be slightly different than the way the immune system responds. So something in this facial cancer allows it to move, allows it to metastasize, to be tumorigenic, because we haven't talked about this, but not every cell grows forever. So most cells die, most cells senesce. The reason that we're not one big tumor is because our cells die. And tumors are bad, are pathogenic, because they don't die in the way they're supposed to, and that they grow in an uncontrolled fashion. So there are two components. You get a cell that essentially the cell line lives forever, and the cell line grows forever and isn't constrained. So there are a set of mutations that lead to those two things. In this devil cancer, what's happened is you've got something that has immortalized the cell line. You've got something that makes it tumorigenic, that ignores the normal constraints on growth, and then something else that makes it look enough like every devil that when that cell gets into the facial tissue of a new devil, it's not immediately destroyed, because most foreign cells are immediately destroyed by the immune system.
The method of action of how it's passed on is really, really fascinating. It's hard to get over the image of these two animals that are fighting. They happen to bite each other and draw blood, and there's this transmission of some cells, and it leads to a face tumor.
Yeah, and the other twist you can put it in, it looks like it's not when they're fighting. It's these two animals having sex, and while they're having sex, they're biting each other. I think we've all been there. But now you're actually transmitting cancer, not just leaving little love marks on your significant other. You've just passed on your cancer.
And do we know, so like we were chatting a little bit about the population change, about that plateau happening. Do we know what any of the genetics are behind the resistant animals, or do we have a sense of how hard they are to work with?
Yeah, so the answer to that is I think it's all too new. So the idea that we have resistant animals, 100% somebody's got to be working on this, but my impression is that this is all new, and this is like the new direction that the research is going in. I don't know how much work has been done trying to understand what makes this cell line so successful. And it seems like that would be an easy question to ask, and it's not, because there's so much genetic diversity out there that identifying the genetic diversity that allows this thing to be tumorigenic and allows this thing to be immortal and allows this thing to be transmitted, that's a lot of questions to try to piece together.
Do we know from chat, do we know if the bites are, if their bites are not in the facial tissue, if the love marks are in the back of the legs, let's say, is that still then a site for these cells to be passed in, or is there something functionally different about the face that they need to colonize originally in? Yeah,
that's a super cool question. Everything that I've ever read just talks about facial lesions, and I don't know that is molecular or biochemical or just behavioral, that this is the way that this species interacts, that they bite across the face. And so we get transmission across that. But that's a really cool question. I mean, again, these are things that we're unlikely to test experimentally, because you're just not going to take an endangered species, bring it into the lab, take a tumor, scratch it on the thigh, and see what happens. But it's an interesting biological question. It's just problematic to study.
So how hard is it to study one of these animals? Because it does seem like they're endangered, right? You can't just it's not like mice where you can ask these kinds of questions, right? And like go in for treatments and like get more of the mechanism of action. Like these kinds of animals seem much harder than our flies and other model systems. Yeah. Do you have any insight on, for the chat, of how one would even go about researching something like this?
There are a lot of questions that we could only answer in the lab, and I don't think we have a lab system for devils. I know we don't have a lab model. I don't know how much breeding. So like endangered turtles, there's a lot of breeding of endangered turtles that goes on in the lab, and they're actually relatively easy to breed in the lab. The issue is, the reason they're endangered isn't the fact that they're hard to breed. It's they're endangered because their habitat is being just absolutely nuked. So with the devils, I don't know if they're hard to breed in the lab. The issue that we're running into is the breeding has been done with these naive individuals, not with individuals that we've just realized they're out there somewhere that are actually more resistant. And those are the individuals that we need to be expanding the population.
Yeah, we pulled up the science paper, and it was demonstrating that they had identified this plateau in about 2020, right? So it's still relatively new in terms of the sciences, and then be able to it was a lot of genomics. So you can imagine like wild catching these animals, doing the genomic sequencing on them, trying to see if they're carriers for the resistance, and then bringing them back and breeding them is not something that's straightforward.
Yeah. And it also happened in 2020. And so everything has been more challenging either in the middle of the plague or coming out of the plague. And that's definitely going to slow things down. I think the encouraging thing is that the individuals are there and that the populations seem to have plateaued. The issue, though, is that they've plateaued at really low numbers. And once you get really low numbers, it doesn't take a whole lot. So what if another pathogen comes through? What if there is, something like an avian flu, but now there's a marsupial flu? So the numbers are small enough that those populations become really fragile just based on a numbers game.
So these populations that have become more inbred than before, the genetic variations lower, and so they're more susceptible to something else coming in.
I just meant the numbers are smaller. And so if you had something that came in and killed off 10% of the population, if you've only got 500 individuals, losing 50 is a bigger deal than if you have 50,000. But you're also entirely correct. As soon as numbers start to plummet, you don't have the opportunity for as much random mating. And so you are going to get inbreeding and drift and potentially have other negative impacts of that, things like greater disease susceptibility.
Right. How does this connect to the species argument? So you were telling us to remember to ask you that near the end. What is the connection with this, and what is a species?
So we sort of touched on this before. I mean, if you believe that there are such a thing as species, then one of the ways that you can identify the species is by their genome. So this species has this genome, this species has this genome. In the case of this immortal parasitic cell line, you now have something with exactly the same genome as a dog, but its phenotype is that it's a tumor. And so that's the twist. Like, is the tumor now a separate species? Because phenotypically it's so different than dog. We normally think of it the other way around, that how different does the genotype have to be for the phenotype to be different? So how different does the DNA have to be for the biology to be different enough that we say they're two different species? Now we have things with very distinct biology, metastatic tumor, dog, and they've got identical genetics.
And yet they are physically very different. So
Different.
So what would you define? I know you don't believe in species, but let's say if we put you on the spot and ask you to define this, is the tumor its own unique thing? And it almost seems more like a parasite now than anything. It doesn't really come across as a dog. It's doing like classic parasitic behaviors, right, for how it needs to pass on its genetic information by infection. But yet it is like lineage wise from 12,000 years diverged from a dog. Is it still dog? At what point would it not be dog?
It's not dog. It's its own beast. And so, by some definitions, I think, and again, I would do this just to ignore the species people, but I think by some definitions, it's now a species. Because the way that I've often gotten around this, so I used to do phylogenetics. Like my PhD was actually on phylogenetic analyses. So phylogenetics is just identifying species. But what I was doing was looking at pairs of genes within a genome. And so I was comparing this gene and this gene, duplicate genes, so copies of genes, instead of this species and this species. Everybody else in the lab worked on species. So they worked on this fish, that fish, this fish, and I worked on this gene, this gene, this gene. The tools that I used were the same thing. So I was saying how related was this gene and this gene by comparing the gene sequences. They were saying how related were these species by comparing the gene sequences. In this case, if we compare the gene sequences, they're identical. If you compare the biology, they're completely distinct. I think by some species definition, a 100% distinct biology means it's a different species.
And yet the genetics are very, very close, if not identical.
No, they're identical. I mean, and so they're not identical, obviously, because there's some genetic change that's created this immortalized cell line. But there's so many genetic changes out there that if you compared how similar or different were two dog breeds, you'd find the same, I think. I'm totally making this up. So straight face or not, totally making this up. But the argument is the same. If you compare two drastically divergent, so compare wolves and Great Danes. They're similar. They can hybridize because they're not species, because species don't exist, right? So they're similar enough that you can exchange genes between these. They're distinct enough that we could identify this is a wolf genome, this is a Great Dane genome. We've got this other genome that's a tumor cell that used to be a dog 12,000 years ago. So is it a species? Because if you're comparing Great Danes and Chihuahuas, they're the same species, but there's a lot of genetic diversity in a Great Dane and a Chihuahua. There's going to be a lot of genetic diversity and a lot of genetic similarity between this tumor lineage and dogs.
So we have a really interesting question from the chat, Thomas. If we eradicate either the dog cancer or the Tasmanian devil cancer, have you eradicated a new species?
Yeah, right. Because and it comes down to the semantics of what's a species, and so we're trying to eradicate various disease vectors like malarial mosquitoes. Is that a good idea? It's a really good idea if you've got malaria, but is it a good idea for the ecosystem? I don't know.
There you go, Sparky Pugwash with the. I mean, I really like the answer because it's one of those where it's like there almost is no real right answer. It's very much like this from the lens of how you're viewing it. And what is that information that you're taking away? And what is, in terms of the definition of the species, why are we doing this? Like in chat, someone earlier mentioned like the differences between the multiple different giraffe species. They can all interbreed. That's the zoos used to just mix them all together. But now for the politics' sake, we cut them into different subgenii or genera so that we can help conserve these groups and these different patterns, right?
Yeah, but it's not for politics. It's because these groups actually are real. So if you have these species, it's because they're genetically isolated. There's something that led to this grouping and this grouping and this grouping. If we artificially then bring them back together, we're destroying a biologically real difference. So the way that we used to deal with this in the world of phylogeny is I would work with just the gene that I was working on, or maybe the species if we were looking at multiple genes. And we would use the cop-out term of operational taxonomic unit. And all this means is it's a sequence on a gene tree. But it's the way that the genes are actually being passed on generation to generation, and we're using phylogenetics just to graphically represent how similar or how related are these genes. So OTUs are real because what we're looking at is change in sequence over evolutionary time. If you want to call it a species, you can, but species is an artifact. It's just a term that we use in some cases for OTUs, lineages of genes that are evolving through time, in some cases for just things to be convenient.
And so next month, we're going to be discussing what is a species.
So let's talk about, so it ends up that just as a bit of a teaser, if you go grab a can of cheap coffee, actually, if you grab a bag of good coffee or a can of cheap coffee, I guarantee you on the side of it, it will say 100% Arabica. And this is so ubiquitous that we now sort of ignore the fact like, okay, so, and the so is there are actually two commercial species of coffee, Arabica and Robusta, or everybody else who's not pretentious just calls it Robusta. So those make up like 96% of the commercial coffee that's out there. Robusta is actually really easy to grow. Arabica is relatively hard to grow. And so at one point, there was a lot of Robusta out there, but it tastes like crap. It tastes, actually, I have limited experience of tasting crap, but I will tell you it tastes like cardboard. And anybody who's inadvertently tasted cardboard, you have tasted Robusta coffee. So
inadvertently has tasted cardboard. I like it.
Okay. So what we'll talk about is what do species mean? What do species mean for your cup of coffee? And what does the fact that So what is the fact that climate change and global change is actually putting at risk Arabica? One of the things that we didn't talk about give me just like 2 minutes, maybe 45 seconds, longer if I keep doing these segues. One of the things that's really frightening with these transmissible cancers is we have created a very unstable planet. And that unstable but globally connected planet is leading to novel disease transmission in a way that this planet has probably never seen. And so one of the reasons that the last plague, the SARS-CoV-2 pandemic, was global was because we were spreading that virus. It's also not clear, as you were talking before, where that particular virus came from, whether it was an animal transmission, whether it was an animal virus that was in a lab that was inadvertently transmitted, what that pattern is. Global climate change is leading to a different disease ecology and a different, in many ways. One is the connectedness, one of it is extreme weather events. All of that plays into an unstable global ecology and unstable disease ecology. So this kind of things with the transmissible cancers, we may be seeing a different landscape of this kind of disease transmission as the global landscape is changing. What we are definitely seeing in coffee is a change of where and when you can grow coffee and what species is actually viable in a particular area. And that's what we can talk about next week. Next month. It'll be sort of cool.
Awesome. Thomas, thank you so very much for your time tonight and chatting about Tasmanian devil face cancer. Just to peel back the curtain, y'all, I'd send Thomas an email. I'm like, are we still good for Monday? I'm excited. And he writes back, yeah, I'm really excited too. Not about getting face cancer, but about chatting. I was like, that's a great email for me. Thank you again, Thomas, so very much. We'll see you sometime next month.
Yeah, and really thank you to the chat. The questions really make this worth my time. It is so much fun to have these conversations.
And I just want to let you know how much folks look forward to our monthly discussions, and it really is a pleasure and privilege to have you. So thank you very, very much for always joining us.
That's awesome. Well, thank you for the kind words. I'll see everybody next month. Take care.
Thomas.